This is the second of a video series on ResearchMaps. This video will be focused on categories of experiments represented in these maps. My name is Alcino Silva and I'm a professor at UCLA Medical School. As I mentioned in the first video, we have recently published a book and an article that you may find useful as additional resources to the material in this video series. What do we need to generate a research map? At a minimum, we would need a strategy to classify experiments, some sort of taxonomy that we could use to organize experiments to be mapped. In addition, we will also need a set of rules to draw these experiments into maps. This is a real map that we derived with the help of an app programmed in the lab. As you can see, the map has phenomena such as GABA inhibition, and connections, either inhibitory or facilitatory, weights that reflect the strength of evidence, and several other things that were already mentioned in the first video and that will be covered in detail in future videos. What would the taxonomy of experiments in neuroscience look like? What kind of categories could we use to classify experiments in neuroscience and then use to map them in our maps? Before we can answer this question, I should stress that neuroscience is highly diverse with many different areas and disciplines, including molecular and cellular systems, behavioral, etc. In this series, we will focus on maps on molecular and cellular cognition. Since experiments in this field involve molecular, cellular, systems, and behavioral cognitive approaches and questions, I believe that what I'm about to describe will be more generally applicable. There are at least 3 different types of experiments in molecular and cellular cognition and in neuroscience in general: identity experiments, tool development experiments, and connection experiments. Identity experiments are not causal experiments. They serve to characterize phenomena that interest us. Connection experiments, on the other hand, test causal connections between phenomena, such as the possible role of a physiological property in behavior. Tool experiments are designed to develop and characterize approaches, techniques, and equipment used to carry out identity and connection experiments. As the figure shows, there are 3 types of connection experiments: positive manipulations, non-interventions, and then negative manipulations. Positive manipulation experiments test a possible causal connection between A and B. In these experiments, we increase or enhance A and then look at B. For example, we would use an optogenetic approach to activate cell type A and then look at behavior B. These experiments tell us that under certain circumstances A is sufficient to trigger B, but by themselves, positive manipulation experiments do not tell us that A normally causes B. Negative manipulation experiments do the opposite of positive manipulation experiments. Instead of enhancing A, they interfere or block A, and then they look at B. For example, we would decrease or even eliminate the activation of a cell type, for example, and then look at the impact on behavior B. Negative manipulation experiments do not tell us that A is normally involved in causing B, since A could simply be something that's needed for B, but it's not involved in actually causing B, such as the relationship between the skull in our heads and learning and memory. Of course, you need a skull to have learning and memory, but the skull is not causally involved in learning and memory. Finally, in non-intervention experiments, we do not manipulate A at all. Instead, we simply track how A and B covary under specific circumstances, such as experiments that would record the activation of cell type A during behavior B. These experiments are critical for assuring us that the relation between A and B, gleaned from manipulation experiments, is not an artifact of these manipulations and instead reflect naturally occurring phenomena. By themselves, however, non-intervention experiments cannot establish causality, since a pattern of covariance could be triggered by a common cause. In other words, the covariance of A and B could simply be the result of the fact that A and B have a common cause but otherwise unrelated. The key is that each of these 3 different types of experiments tell us something very different about the possible relation between A and B, and thus their combination is especially powerful at elucidating the relation between A and B. We will cover this idea in more detail in the next video of this series. The connection experiments we just described are single connection experiments, but in addition, we have multi-connection experiments. Multi-connection experiments are usually carried out for mediation analysis, in other words, for determining the mechanism or the possible mediators of a causal connection. For example, how would we go about testing the role of calmodulin kinase 2 in mediating the effects of NMDA receptors on CA1-LTP? One strategy will be to increase the levels of NMDA receptors, which would be expected to enhance CA1-LTP and at the same time block chymogene kinase 2. We would expect that the block of chymogene kinase 2 would also block CA1-LTP. However, if chymogene kinase 2 did not mediate the effects of NMDA receptors on CA1-LTP, then we would expect that CA1-LTP would be left unaffected by the block of chymogene kinase 2. This really is the point of multi-connection experiments. They are designed to manipulate more than one phenomena so that their relations are elucidated. Finally, we will talk about identity experiments designed to characterize individual phenomena. There are at least 3 different types of identity experiments: what experiments, where experiments, and when experiments. What experiments are designed to define the composition of an entity and its association with other entities. Where experiments are designed to specify the location of an entity relative to appropriate landmarks. For example, neuroanatomical experiments that define where a channel is expressed. When experiments define the developmental stage or any other timeline relevant to that phenomena. For example, when experiments will find out whether a channel is expressed in development or in the adult. This figure summarizes what we have covered in this video. However important, dividing experiments into categories is not sufficient to generate a map. In addition, we also need a set of rules for drawing these maps. This will be the focus of the next video in this series. This brings us to the end of video 2 in this series dedicated to neuroscience research maps. I hope you found this video informative, and I encourage you again to consult the book and paper that were mentioned in the beginning of the video. Also, I stress again that although we believe that the principles covered in this and other videos of this series will apply to many areas of neuroscience, it is possible that they would be insufficient to adequately map other areas, such as, for example, systems neuroscience. We need system neuroscientists, cognitive neuroscientists, etc. to help us develop the frameworks and rules required to effectively map experimental findings in these other areas. I see this as a community effort, one that will dramatically change how we review the literature, carry out experiments, and teach science.